chance to present the work today. So switching to something completely different, so we are talking about Salmonella ST34, or more commonly known as Salmonella typhimurium monophasic variant, and also about multi-drug resistance. So it's all about bad news, I think. So actually, the investigation actually started from diarrhea surveillance study we conducted a while ago, from 2014, and where we recruited more than 3,000 children with acute diarrhea. And the initial plan was to look at the prevalence of cirrhosis in the Vietnamese population. However, by the end of the study, we actually see more cases of Salmonella than cirrhosis. At the end, around 15% of cases were infected by Salmonella, and when we subjected all of those for whole genome sequencing, and the first aim of the study was to use genomics to look at the diversity of serotype and AMR factor in the Salmonella. And then we found that ST34 was the predominant serotype, and it has a high level of non-susceptibility to ceftriaxone, to ciprofloxacin, and to azithromycin. And also, the MDR status was correlated with prolonged hospitalization, so it was clinically important. And however, in this talk, I must focus more on how we use genomics to understand the local and the global evolution of this ST34. So I think the audience probably need no introduction to Salmonella anyway. However, the variant that we're focusing here today is ST34. So it emerged out of the pig population in Europe in 2005, and then rapidly spread into cattle, poultry, and then human internationally. And it has become the most widespread and most prevalent Salmonella serotype identified worldwide. So compared to the common typhimurium, ST34 differ in the deletion of the FLIP-B antigen, the flagellar antigens, leading to the monophasic serotype, and also is usually resistant to some conventional antibiotic, including streptomycin or ampicillin. ST34, even though it causes diarrhea with mild symptoms, sometimes is quite important because they are the culprit for multiple foodborne outbreaks. And the one shown here on the slide is the 2022 Kinder chocolate outbreak occurring in Europe, where you remember that the ST34 sent 450 children to the hospitals, and also caused a recall of more than 3,000 tons of chocolate worldwide. So it's a huge economic burden. Okay, so our research questions were to look at the population structure of this ST34 in Vietnam, and also to look at the genetic determinants of AMR in this variant. And to do that, we compiled a collection of more than 450 genomes of this variant, mostly coming from Asian setting, because the Salmonella is like very widely, intensively studied in high-income setting. However, in Southeast Asia, it's kind of a neglected topic. So most of the isolate coming from the two studies we conducted in Vietnam, the one that I mentioned to you previously, the pediatric population, and another one that we published previously on the Salmonella causing bloodstream infection in HIV patients. Okay, so a little bit complicated slide. So shown here on the left is the time phylogeny of this ST34 at the global level. And isolation, hopefully you can see the color. The color of the tip is labeled based on the source of isolation. The Vietnamese isolates were highlighted in the boxes, and they, you can see they represented at least 5 major introductions into Vietnam. And if you look at the TMRCA, that means the time to most recent common ancestor, or the time of introduction of these into Vietnam, they were quite in the short window time frame, about from 2003 to 2006. 2003, 2008. And notably, for each of these clusters, if you look at the heat map on the right, the sustained transmission or the clonal expansion of these clusters were linked to the carriage of either a multi-drug resistant plasmid of in AC or in HI2. And for example, in the VN2 cluster, you can see that they're carrying the plasmid in AC that co-transferring plasticity and 55, QNIS1 and MCR 3.1. So that means co-resistant to ceftriaxone, to quinolone, and also to the colistin. So in order to understand more about the plasmid, we opted to use number 4 sequencing. And here is the structure of the plasmid. So I'm not going to bore you with all the comparative genomic. But one important thing to take home from this plasmid is that even the size is relatively modest. The plasmid carry 11 AMR genes and conferring resistance to conventional and also clinically important antimicrobial, including Streptococcus. Another interesting thing about the plasmid, you focus on the green part, the green element, is that they're also carrying regions of transfer region that allow the plasmid to be conjugative, and then we have confirmed that in the lab. So that's also probably explain why the plasmid is also found in some isolated in China and also from Australia. So there's some potential for the plasmid to be transferred into all the different clones. And the next step that we took was to investigate that in more than 10,000 of publicly available SD34 genomes and selected the one that either positive for CTSN55 or for MCR3.1. And then we added that into the existing origin. So more trees, but this time is a little bit simpler. So if you only focus on here, so here are 3 different clones that have the stable inheritance of the plasmid, and you look at the one that is circled in black, that means the newly added isolation. And you can see, even for example, for the clone can called VN2, the clone evolved and transmitted in Southeast Asia, but also there's some instances of transfer or exportation of the bacteria SD34 to the US, to Australia and China. And for example, on the right you can see the Australian lineage 1, even though it's called Australian lineage 1, but if you look at the root of the tree, you can see that they are isolate coming from either Vietnam or Thailand. So it emerged and evolved in Southeast Asia and then spread to high income setting. Okay. So these data suggest that the SD34 probably use Southeast Asia as a hub for transmission for worldwide success. So we opted that to use some more sophisticated methods like stochastic mapping to infer the transmission of these isolates, and found that aside Europe, Southeast Asia also serves a major source of transmission of SD34 worldwide. And the picture on the right just to show you that we also ran a lot of randomization, chip location randomization to actually confirm that the signal is true. Probably stop talking now. And just to ensure that actually the phylogeographic signal was and I think the key take-home message that SD34 success transmission is dependent on the carriage of NDR plasmid, and also Southeast Asia should be viewed as an increasingly important hub for transmission of this pathogen. And also continue to monitoring the emergence and spread of potentially XDR plasmid in non-typhoid salmonella. And with that, I want to extend my thanks to the team in OCRU, especially Dr. Yui and his team, also the collaborator we have in Vietnam, also in the UK, and also to welcome Trust for funding the study. Thank you very much.
Any questions?
ICMC 3.1 in VN4. How about the mechanism of the mixed antimicrobial resistance in VN4?
So not VN2, right? VN4. Yeah. So the mechanism of resistance for the VN4. So just to point out, the VN4 is different clone. So we only focus on VN2 because the shortage of time. But VN4 also carries an AC plasmid, different in AC plasmid compared to the VN2. They're coming from 2 different parts of the tree, and VN4 is not conjugative. So the plasmid itself, it could not be transferred.
All right. This expression can be imply or predict the severity or predict only the channel resistance?
in terms of severity for patient, because we don't treat patient with colistin. And also MCR, in the literature, usually MCR doesn't confer full resistance to colistin. So I think the MCR present here is sounding the alarm that it could be spread into all the gram-negative. That can cause more severe infection. Also, it can point to that the source of the origin for these AMR plasmid coming from the animal setting.
Before your expansion or sequencing, do you think the helpful for this expression or sequencing is physics or blood?
You mean to extract the
Specimen.
The specimen. So here actually we had two different specimens. So the one coming from VN2, I'm talking mostly from isolated from Vietnam. That VN2, 3, VN1, and VN4, they're coming from a diarrhea stool sample from children. And the one called VBSI is the one coming from blood from patients. So we isolated the bacteria from bloodstream infection in HIV patients.
Thank you.
Thank you very much for your questions.